Welcome to ClassMate. I'll help you learn immunology. In this comprehensive overview, we're going to explore the incredible systems your body uses to protect itself from invisible threats. From the physical barriers of your skin to the complex cellular armies patrolling your blood, prepare to be amazed by your own biology. Every single day, your body is under constant siege by a staggering variety of microscopic invaders. Bacteria, viruses, and fungi are everywhere in our environment, constantly looking for a way in. Fortunately, you are equipped with an incredibly sophisticated defense system that works around the clock to keep you safe. Your first line of defense is your physical and chemical barriers. The skin is a nearly impenetrable wall, while your mucosal surfaces use chemical secretions to trap and neutralize pathogens before they can even enter your tissues. It's a remarkably effective gatekeeping system that prevents most infections. Once a pathogen breaches those initial walls, it encounters the patrolling units of your innate immune system. Within your blood vessels, a vast standing army of white blood cells, or leukocytes, is constantly on the move, monitoring every corner of your body for signs of trouble or invasion. The innate immune response is rapid and nonspecific. One of its key players is the macrophage, a powerful cell that identifies invaders and literally eats them. Through a process called phagocytosis, the macrophage engulfs the bacterium, breaking it down and neutralizing the threat almost instantly upon contact. How do these cells know what to attack? They use pattern recognition receptors, like toll-like receptors, to identify conserved molecular patterns found on microbes but not on human cells. These PAMPs act like universal danger signals that tell the immune system exactly when an intruder is present. Another vital part of the innate response is the complement system. This is a cascade of proteins that can assemble into a lethal structure called the membrane attack complex. This complex literally punctures holes in bacterial cell walls, causing them to burst and effectively destroying the invading pathogen. When tissue is damaged or infected, the body triggers inflammation. You'll see blood vessels widen and become more permeable, allowing more defensive cells to rush to the scene. This process, known as leukocyte extravasation, ensures that your immune forces are delivered exactly where they are needed most. Natural killer cells specialize in identifying host cells that have gone rogue either due to viral infection or cancer. They scan for missing self signals, such as the absence of MHC class I molecules. If a cell looks wrong, the NK cell triggers its destruction to protect the rest of the body. The lymphatic system acts as the communication highway of your immunity. This vast network of vessels and nodes allows immune cells to travel and share information. It's here, in the lymph nodes, where the innate system hands off vital intelligence to the even more specialized adaptive immune system. Adaptive immunity often starts with a dendritic cell. These cells act as scouts, capturing antigens at the site of infection and carrying them to the nearest lymph node. This migration is crucial as it brings the evidence of an invasion directly to the specialized cells waiting to respond. Inside the lymph node, the dendritic cell presents the captured antigen to T cells. The T cell receptor must perfectly recognize the MHC peptide complex presented by the scout cell. This precise molecular handshake is the key that unlocks the powerful and highly specific adaptive immune response we're about to see. Activation requires 3 distinct signals to prevent accidental responses. First is the antigen recognition, second is co-stimulation, and third is the influence of specific cytokines. This multi-step process ensures that the immune system only launches its full-scale offensive when it is absolutely certain an infection is present. Once activated, the T cell undergoes clonal expansion. It begins to divide rapidly, creating thousands of identical copies of itself in a matter of days. This sudden surge in numbers transforms a single specialized cell into a massive population of effector cells ready to seek and destroy the invader. CD8 plus cytotoxic T cells are the frontline warriors of the adaptive system. They identify and latch onto infected cells, releasing lethal proteins called perforin and granzymes. These molecules force the target cell to undergo apoptosis, or programmed cell death, effectively stopping the viral replication or cancerous growth in its tracks. CD4 plus helper T cells act as the generals of the immune system. Instead of killing directly, they secrete chemical messengers called cytokines to coordinate the entire response. These signals activate B cells to produce antibodies and empower macrophages to kill more effectively, ensuring all branches of immunity work in harmony. B cell activation is equally complex, requiring the binding of an antigen and assistance from a helper T cell. This cognate help ensures that B cells only produce antibodies against confirmed threats. Once the signal is received, the B cell is ready to transform into a specialized factory for immune defense. The activated B cell matures into a plasma cell. These are essentially biological factories capable of pumping out thousands of Y-shaped antibody molecules every single second. These antibodies circulate throughout your body, seeking out the specific antigen they were designed to recognize and neutralize with incredible precision. There are 5 classes of antibodies, each with a unique structure and role. From the abundant IgG that provides long-term protection to the secretory IgA in your mucosal linings, and the specialized IgE involved in allergic responses. Each class is a specialized tool optimized for different environments and threats. Antibodies work through several mechanisms. They can neutralize toxins by blocking their active sites or coat bacteria in a process called opsonization, which acts like a glowing beacon for phagocytes. These actions make it much easier for your innate cells to clean up the remains of an infection. One of the most remarkable features of adaptive immunity is memory. After an infection is cleared, long-lived memory B and T cells remain in your body for years. If the same pathogen ever returns, these cells mount a response so rapid and powerful that you may never even feel sick. However, the immune system isn't perfect. Sometimes it overreacts to harmless environmental triggers, leading to hypersensitivity. Whether it's pollen, dust, or certain foods, these reactions cause the body to launch a full-scale immune response against something that poses no real threat, leading to the symptoms we call allergies. Type 1 hypersensitivity is the classic allergic reaction. It involves IgE antibodies that coat specialized mast cells. When an allergen binds to these antibodies, the mast cell releases histamine in an explosive process called degranulation. This sudden release is what causes the itching, swelling, and redness we associate with allergies. Type 2 hypersensitivity occurs when the immune system mistakenly targets the body's own cells. Antibodies may bind to antigens on the surface of healthy red blood cells, for example, marking them for destruction by other immune cells. This misguided attack can lead to serious conditions where the body destroys its own blood. In type 3 hypersensitivity, the problem lies with immune complexes. These are clumps of antibodies bound to antigens that circulate in the blood and eventually deposit in vessel walls or tissues. Once trapped, they trigger a localized inflammatory response that can cause significant damage to organs and blood vessels. Type 4 hypersensitivity is a delayed response mediated entirely by T cells. Unlike immediate allergies, this reaction takes 24 to 48 hours to develop. A common example is the skin rash caused by contact with poison ivy, where T cells migrate to the area and trigger a persistent itchy inflammation. Autoimmunity represents a fundamental failure of self-tolerance. In conditions like multiple sclerosis, the immune system mistakenly identifies the body's own tissues as foreign. Here, T cells attack the protective myelin sheath surrounding neurons, disrupting nervous system signals and causing a wide range of debilitating clinical symptoms. On the other hand, immunodeficiency occurs when the immune system is weakened or non-functional. This leaves the body dangerously vulnerable to infections that a healthy person could easily fight off. These gaps in our defenses can be caused by genetic factors or acquired through certain diseases and treatments. Primary immunodeficiencies are usually genetic. In some severe cases, the body is born without the ability to produce functional B or T cells at all. Without these vital adaptive defenders, even the most common environmental microbes can become life-threatening, requiring strict isolation and advanced medical interventions. Secondary immunodeficiency is acquired later in life. The most well-known cause is HIV, a virus that specifically targets and destroys CD4 plus helper T cells. By depleting the generals of the immune system, the virus effectively dismantles the body's ability to coordinate any meaningful adaptive defense against pathogens. Without a functional immune system, opportunistic infections take hold. These are pathogens that rarely cause disease in healthy people, but are devastating to those with immunodeficiency. Fungal pneumonia, for example, can spread unchecked in the lungs when the cellular immunity required to contain it is no longer present. Transplantation immunology deals with the challenge of foreign organs. Because every person has unique MHC markers, your immune system may identify a transplanted kidney or heart as an invader. This leads to organ rejection, where your T cells attack the new tissue unless suppressed by specialized medications. We are now entering the era of cancer immunotherapy. Scientists can now engineer a patient's own T cells with specialized receptors called CARs. These CAR T cells are designed to specifically identify and destroy leukemia cells, turning the patient's own immune system into a powerful weapon against cancer. Monoclonal antibody therapy is another breakthrough. We can create synthetic antibodies in the lab to target specific molecules in the body. For example, these antibodies can neutralize pro-inflammatory proteins like TNF-alpha, providing relief for millions of people suffering from chronic autoimmune conditions like rheumatoid arthritis. Vaccinology is one of the greatest triumphs of medicine. By introducing harmless fragments of a pathogen, like mRNA or protein subunits, we can prime your immune system. This allows your body to build a library of memory cells, providing you with powerful protection before you ever encounter the real disease. Immunology even plays a role before birth. Through passive immunity, a mother transfers protective IgG antibodies across the placenta to her developing fetus. This provides the newborn with essential immune protection during their first few months of life, while their own immune system is still developing its capabilities. In the lab, diagnostic immunology allows us to measure your immune health. Tools like the ELISA test use color changes to quantify specific antibodies in your blood. This helps doctors diagnose infections, monitor vaccine responses, and understand the status of various autoimmune or inflammatory conditions. Flow cytometry is another powerful tool for immunologists. It uses lasers to analyze and sort thousands of individual cells per second based on their surface protein fingerprints. This allows us to precisely map the population of immune cells in a patient, providing invaluable data for research and clinical diagnosis. The future of immunology is incredibly exciting. With gene editing tools like CRISPR, we are moving toward curing genetic immunodeficiencies at the source. By repairing mutations in stem cells, we can potentially give patients a brand new, functional immune system, opening the door to treatments we once thought impossible. We've covered a vast landscape of science today, from the first line of defense to the frontiers of gene editing. Immunology is a journey of discovery that never truly ends. Thank you for choosing ClassMate. See you in the next lesson as we dive deeper into each system.